Lecture 1, Enzyme Regulation, Feedback Inhibition versus Enzyme Synthesis. Enzymes are the catalyst of metabolism, and their regulation is vital for controlling metabolic flux. There are two broad categories of enzyme regulation. 1. Feedback inhibition, allosteric regulation. A rapid, reversible control mechanism. The end product of a metabolic pathway inhibits an enzyme acting earlier in the pathway. Common in biosynthetic pathways. Example, threonine deaminase inhibited by isoleucine in amino acid synthesis. Mechanism. Involves allosteric binding sites distinct from the active site. Binding causes conformational change, reducing enzyme activity. Advantages. Fast response to changing cellular needs. Does not require new protein synthesis. 2. Regulation via enzyme synthesis, gene-level control. A slower, long-term regulation strategy. Involves transcription and translation of genes encoding metabolic enzymes. Controlled by inducers, repressors, and activators. Example. Glucokinase gene expression is upregulated by insulin in the liver. Enzymes involved in gluconeogenesis are induced during fasting. Lecture 2. Catabolite repression and product repression. In microbial systems, especially E. coli, certain global mechanisms regulate the preference for energy sources and pathway utilization. 1. Catabolite repression. This mechanism ensures that glucose, the preferred energy source, is utilized before others, like lactose or arabinose. Key elements. CAMP, cyclic AMP, levels are low in the presence of glucose. Low CAMP results in reduced activation of alternative catabolic operons, e.g., lac-operon. CAP, catabolite activator protein, requires CAMP to activate transcription. Process. Glucose inhibits adenylate cyclase. CAMP decreases CAP inactive lac-operon not expressed. When glucose is depleted, CAMP increases CAP binds transcription of alternative sugar operons racemase. 2. Product repression. When the end product of a pathway accumulates, it represses the synthesis of enzymes involved in its production. This regulation often occurs at the transcriptional level. Example. Tryptophan operon. High tryptophan levels lead to repression of tryptophan biosynthesis enzymes. Lecture 3. The lactose operon, lac-operon. The lac-operon in E. coli is one of the most studied models of gene regulation. 1. Structure. Structural genes, LACs, LACI, LACA. Promoter and operator regions. Regulator gene, LACI, encodes the repressor protein. 2. Mechanism. In the absence of lactose, the LAC repressor binds to the operator blocks RNA polymerase no transcription. In the presence of lactose, lactose or allolactose binds to the repressor conformational change. Repressor detaches, transcription occurs. 3. Glucose affects catabolite repression. Even when lactose is present, if glucose is also available, the operon is not fully activated due to low CAMP. This dual regulation ensures energy-efficient metabolism. Lecture 4. The arabinose operon, ERA operon. The ERA operon controls the utilization of arabinose, another sugar. 1. Structure. Genes, ARAB, ARA, AROD. Regulated by the RSC protein, which can act both as a repressor and an activator depending on conditions. 2. Mechanism. Without arabinose, RSC binds to distant sites, forming a loop that prevents transcription. With arabinose, RSC binds arabinose changes conformation binds to adjacent sites facilitates RNA polymerase binding transcription starts. This operon is another example of dynamic transcriptional regulation influenced by nutrient availability. Lecture 5. Identification of different regulatory mechanisms. Researchers identify metabolic regulatory mechanisms using a variety of tools and approaches. 1. Genetic approaches. Gene knockouts observe changes in metabolic flux or phenotype. Reporter genes link regulatory regions to easily measurable outputs, e.g., GFP or beta-galactosidase. Mutagenesis, random or targeted mutations to discover regulatory elements. 2. Molecular techniques. Northern blotting, qPCR, measure mRNA levels. Western blotting, quantify protein expression. Chromatin immunoprecipitation, CHIP identifies transcription factor binding sites. 3. Metabolomics and systems biology. Mass spectrometry and NMR identify and quantify cellular metabolites. Flux balance analysis, FBA, estimates pathway activity based on stoichiometry. Transcriptomics and proteomics show how changes in gene and protein expression affect metabolism. These approaches help map regulatory networks, especially in metabolic diseases or biotechnological applications. Lecture 6: Systems-level regulation of metabolism. Beyond single pathway control, organisms regulate metabolism at the systems level through feedback loops, hormonal coordination, and tissue specialization. 1. Feedback loops. Operate across levels, enzyme activity, gene expression, and hormonal signaling. Example: high ATP levels inhibit phosphofructokinase in glycolysis. 2. Tissue-specific regulation. Liver, gluconeogenesis, urea cycle, cholesterol metabolism. Muscle, glycogen storage and glycolysis. Adipose tissue, lipogenesis and lipolysis. Brain, glucose-dependent, ketone utilization during fasting. 3. Circadian and environmental control. Metabolism varies with the day/night cycle. Environmental cues, light, food intake, temperature, synchronize metabolic rhythms. 4. Disease and dysregulation. Metabolic disorders, e.g., diabetes, metabolic syndrome, involve breakdown in regulation. Cancer cells rewire metabolic pathways, Warburg effect. Viral infections, e.g., COVID-19, can hijack host metabolism. Understanding these patterns enables the design of therapeutic interventions, precision nutrition, and synthetic biology applications. Module 2 Summary. In this module, we explored enzyme regulation mechanisms, feedback inhibition and transcriptional control, global repression mechanisms, catabolite and product repression, operon systems, lack and error operons, techniques for identifying metabolic regulation, systems biology and tissue-level metabolic control. With this knowledge, you now have a solid foundation for understanding how metabolism is controlled at molecular, cellular, and systemic levels. These concepts are essential for biochemistry, medical research, biotechnology, and nutrition science.